a living, breathing thesaurus sitting next to me in the studio. So tell us Orna's founding story. What got things started there?
Yeah, so Orna has a very traditional start in the sense of if you go back to how biotech companies used to go. So it used to be that academics would discover something and they would start a company and pull some sort of nascent team together and then try and get venture interested. Now, of course, these days, venture typically goes and scours science directly from universities and so on and builds the company to their own sort of preconception. But Orna was really in that old mold. So our story goes back to 2016 or 2017. Dan Anderson of MIT is listening to a talk on naturally occurring circular RNA in China and goes, I wonder if that could be an interesting variant on a theme. Of course, Moderna already existed at that time. 11 was coming through, and it has already been known for attempting at least the RNA-based editing. And Dan had a long history in LNPs and RNA in any case. And so he gave the task to a graduate student, Alex Wesselhoft, who completely blew it out of the water. PhD finished in 3.5 years, green thumbs at the bench, and solved the fundamental problem, which is how can you make circles efficiently at scale and large enough to be useful, and figure out how to make them useful by getting them to express things. So he did all that. And so that was the basis of IP that was filed. And then they thought, well, there should be some value here. And so the company, a company was formed. It wasn't called Orna at the time. And they basically encountered MPM Capital, it was known now, MPM Bioimpact. And this is in the 2019. So the first funding came in mid-2019, but part of that funding was a conception that what this company should do with its circles is to see if it could figure out a way of doing an in vivo thing, but not editing at this time because the RNA is going to be transient. But can we get the RNA into immune cells? And if we could we disrupt or displace CAR Ts and cell therapy completely because it would be so much more convenient? So in other words, there's a very high science concept that predates me, and it was really an idea that came from MPM. And so money came in and they started building the team immediately with Alex on board. And I started talking to MPM. I had left Intellia early in 2019. I was scouting about for things to do, and I started talking to them in October or so. And we kind of sealed the deal in December. And so I started in early 2020.
Mm-hmm.
And you had no idea that the world was about to blow up and become all RNA all the time.
Exactly. No,
it was fated.
I like to think I had a crystal ball, but I did not.
Nobody did.
Yeah.
It is all RNA all the time. And it's like, and how many letters now precede RNA across the spectrum?
Quite a few.
There's quite a few.
Letters, shapes.
Coding, non-coding.
Well, that's one of the things I think that's cute about the term we invented for what we do. You know, something that I noticed early on is that up until that time, certainly in the papers that Alex and Dan had published, and in the early materials, it was called circRNA because CIRC, lowercase, because that's what it's called. Mm-hmm. That's the standard abbreviation. But I realized almost immediately that people get confused. They look up circRNA, and there's thousands of papers. There's a lot of papers on circRNA, but none of that is relevant to what we're doing because ours is completely synthetic. We're not extracting circRNA from cells. We're making it in vitro, designed to encode exactly what we want, and it's made in a different way than it's made in vivo. It just in the end looks like a circle. So we need a branding for what this is. And I remember being at a bar one Friday, as one is, and
it's where all the great ideas come from.
Exactly, thinking of a name of the company as well. Actually, really, that's what I was trying to do. And I thought, Moderna's kind of cute because it's like mode RNA, and their abbreviation mRNA.
Mm-hmm.
And I thought, we need something. What could be RNA? Is there an RNA something we could call the company? And I thought, well, I don't know. RNA. I thought, that's cute, because then we could call it also oRNA, where the O just means circular. It doesn't stand for an O word. It just means circular. And so that was just born in a flash there. And so we changed.
The important question is, what were you drinking at the time?
It was probably a beer. But so that's when the name came in. So that was in That was shortly after I started there.
Yeah. I'm going to ask both of you guys to unpack for me a little bit the advantages, differences between circular RNA, oRNA, and specifically in Orna's case, synthetically produced oRNA from. And we could go long on this conversation. I don't want to go too long. This is the business of biotech. It's not the science of biotech. But for our listeners' benefit, maybe just more color on where oRNA fits in the landscape that we've seen linear RNA for quite some time.
Yeah. Well, the thing that intrigued Dan back in the day was that even to this day, people don't really know what naturally occurring circles do. There's a number of theories, but to my instinct, I don't think any of the theories have really nailed what they're there for. Doesn't quite feel right. But the one thing that people know is that naturally occurring circular RNAs hang out in cells a lot longer than the linear RNA counterparts, particularly especially if it's derived from the same gene. The circular piece is more long-lived than the linear piece. So circles live longer than lines. And so that was really what Dan said. Well, okay, if you could do a whole technology based on circles, it would be better. Longer half-life is better. And so we've gone on to show that that's true, and I think it's been shown now in multiple people's hands. But what we discovered along the way is that circular RNA, I call it the gift that keeps on giving, because it continued to show these unexpected advantages over mRNA. And the way I would summarize it, again, coming back maybe to the business and less on the science, is that if the world could make circular RNA the way we make circular RNA for products, nobody would make linear RNA products anymore, because circles have advantage upon advantage over lines and no disadvantages. There's no trade-off. There's basically fewer, better, more bits and pieces to get a circle to work than there is for a line. So it's funny because, of course, the mRNA is our RNA. It's how instructions are encoded. So naturally, we try and mimic that in the product, and no one knows what circles do. But as a biotechnology, as a product, the correct answer is circles. It's not the way we do it, but the correct version of a product should be a circle because it's more stable. It's easier to make. It's far cheaper to make. It's made at high yield. You can make them much larger. You can express them much higher. They hang out in cells much longer, and they package much more efficiently in the lipid nanoparticle at the end of the day. So they're just better in every way.